Welcome to the Allen Institute. Today we'll be taking you behind the scenes of the PatchSeq pipeline, the process by which we collect multimodal data from individual neurons and slices of living brain tissue. PatchSeq combines patch clamp electrophysiology, a technique to record the electrical activity of the cell, RNA sequencing, which tells us what genes are expressed by the cell, and morphology, the three-dimensional shape of the cell, which we obtain through imaging and digital reconstruction. Gathering all three types of data for each neuron allows us to investigate how these properties relate to each other and serves as a valuable reference for the neuroscience community, almost like a Rosetta Stone for translating between the languages of 3D structure, electrical activity, and transcriptomics. As you'll see, it takes a whole team of researchers working together to create this extremely standardized, high-throughput pipeline for sample preparation, data analysis, and data collection. Let's start in the tissue processing lab, where a brain sample begins its journey.
So today we're working with some mouse tissue. In this lab we do work with human and non-human primate tissue as well, but today the PatchSeq team is going to be working with mouse, so that's what I'm preparing for them. Basically we just have this chuck that we put the brain onto with some super glue. So I plop it right in here, get some artificial cerebrospinal fluid so that the brain's happy, and I'll make sure the brain also has a oxygenated gas that we feed the line to here. So see a little bubbles coming out. So this is like a vibrotome. It allows us to get really fine precision slices. We're going to be slicing this brain at 350 microns. So it's like a glorified charcuterie machine where we can get extremely thin slices every single time. So once I get a slice coming off the vibrotome here, I can pick it up with my transfer pipette, and I have to bring it over to our incubation machine here. This allows the slice to be kept at the same body temperature as most mammals, and we leave the slices in there for about 10 minutes. So after the slices are done incubating, we can take them out and put them in this room temperature holding bath. That is another version of artificial cerebrospinal fluid. And once they're all in this bath, we can bring them out to the physiology team.
Welcome to the Integrative Cell Physiology Lab, and this is where we do PatchSeq experiments. Today we are working with mouse slices, and these are transgenically modified mice that express a fluorescent reporter in a particular cell type. So we use this to guide our targeting for our PatchSeq experiments. Here we have one of our 8 patch clamp electrophysiology rigs, and this is where we are able to take a fine glass electrode that is pulled to the width of a neuron. And she's loading that glass pipette down to what we call the chamber. And within that chamber is our mouse brain slice. And so this is where the patching process begins. You can see that she is navigating that glass pipette through the tissue, and there's a bit of positive pressure that's being expelled through that pipette, and it's moving the tissue as she finds the cell to patch. So here's the cell that she's going to patch. She's going to move the pipette and just touch the surface of that neuron and then rupture the little piece of the membrane that's under the tip of that pipette. And that is what gives us electrical and physical access to that neuron. Once we've broken into the cell, we can apply a series of current steps where we can then look to see how does the cell respond? What is the shape of the action potentials? How does the cell fire? And here you can see a current experiment where looking at the red trace, we can see what the voltage response of the neuron as we apply different current steps, and you can see this neuron is firing action potentials, and then we can take a quantitative approach and measure some of the properties of those action potentials firing. So during the PatchSeq process, we are also filling the cell with a dye, and what you can see here is a fluorescent image of the dye that is now being diffused throughout the neuron. So this allows us to get the morphology of the neuron or the shape of the neuron, and then we can relate that back to the electrophysiology and the transcriptomics and really get a unified view of what the cell type is. So at the end of the PatchSeq experiment, we apply a small amount of negative pressure through that tip of the pipette where we're able to collect some of the RNA from the cytosol, but as well as the nucleus. And you can see right here is on the tip of the pipette is the nucleus that we have just extracted from the neuron. And then we can take that nucleus and deposit it into a lysis buffer and send it to our molecular biology group, where then they can do PCR and understand what genes the neuron expresses. So here at the Allen Institute, we have standardized our PatchSeq pipeline where we have each rig operator can collect 5 to 6 experiments per day, and we can multiply that across 8 rigs. And then that gives us upwards of collecting around 3,000 to 4,000 neuron PatchSeq experiments per year.
Hello everyone, welcome to the Histology Lab. So we receive the slices or the pieces of tissue from the physiology team, and we stain the tissue sections in a 3-day protocol in order to enhance and visualize the neurons. Here my colleague is mounting the tissue section on a regular slide. And when the slides are ready and dry, we pass them on to the imaging team.
So the slides have been mounted and stained by histology, and now they've been brought up to the imaging lab. The first step in the process is we do an overview scan. So what we do is we take the slides and we load them into a slide tray like this, and we're going to put them into the slide scanner, which is going to kind of autonomously scan all of these slides at a low magnification and get the entire tissue. Once these low magnification scans are complete, we will send them to morphology, and they will let us know which cells on the tissue they would like to reconstruct. And once we know that, we're going to move over here and we're going to image the tissue at a high magnification. So the high magnification scans are done at 63x, and the reason we need that high magnification is the neurons are very, very small, and we need to be able to capture all those tiny little twists and turns that the dendrites and the axon are doing throughout the tissue. The scan's now running. Once it's done, we will send it to morphology, and then they will proceed with the reconstruction.
Welcome to Morphology. Our goal is to create accurate representations of different neuron morphologies within the brain and in different species. From the imaging lab, we receive both a 20x overview image as well as a 63x image stack. On the right side of my screen is our 20x overview image containing a single slice of specimen tissue with each of the cells that were patched labeled on it, which provides us with context as to where the cells are located within the brain. On the left of my screen is our 63x image stack, which shows one of the individual cells at higher magnification. We import the 63x image series into our reconstruction software. And here we're able to visualize each of the 2D images in a 3D image stack. So here I'm zoomed in on the soma, and we can see where each of the dendrites branch from the soma, as well as where the axon branches down here. And we can see here is the reconstruction. So in pink is the apical dendrite, blue is the basal dendrite, and red is the axon. We create our reconstruction by placing nodes in 3D space. So as I zoom in on this branch of untraced dendrite, I'll create the reconstruction by placing nodes along the 2D images where the signal is the darkest. So from this, we connect our different branches together, which then allows us to get our full reconstructed neuron. From our reconstruction, we also gain information such as axon length, number of axon branches, and the number of dendrites exiting the soma. This allows us to quantitatively describe different types of neurons located within the brain.
Ultimately, these morphology reconstructions are combined with the electrophysiology data and the transcriptomic data, along with anatomical information as to where the cell was located in the brain. And that's all packaged into data sets that we share freely with the scientific community. This PatchSeq dataset is being used here at the Institute to classify thousands of unique cell types in the brain, to investigate evolutionary differences across species, to discover which cells are affected by diseases, and more. Our data is also being used by neuroscientists around the world to answer all sorts of other questions, and you can be one of them. Visit brain-map.org to find this PatchSeq dataset and lots of other open neuroscience resources. Happy exploring!